I'm Camila Mazzoni, and I'm here to give you a brief overview about the European Reference Genome Atlas of Biodiversity, the ERGA consortium. Europe has about 200,000 species of eukaryotes recognized. Out of the European species that were assessed by IUCN, almost 1/4 is considered as threatened with extinction. Europe has recognized the biodiversity crisis and has adopted an ambitious and long-term plan to protect nature and reverse degradation of ecosystems, the EU Biodiversity Strategy for 2030. The first steps of the ERGA consortium happened during pandemics, when scientists worried with the biodiversity crisis, but also motivated by the latest advancements in genomics and also existing genome projects, got together to discuss the possibility of creating a pan-European biogenome project. The ERGA consortium grew fast and established a connection early on with the international Earth BioGenome Project, becoming the European node of EBP, aligning goals with the international consortium and also aiming at contributing to the mission of characterizing high-quality genomes of Earth's eukaryotic biodiversity. The workflow to generate high-quality genomes is multidisciplinary. It ranges from fieldwork and taxonomic expertise to molecular biology applications, bioinformatics, and computational tools. This range of expertise is well represented in ERGA in the more of 700 scientists that are part of the consortium, belonging to 40 European and associated countries. With the complexity of working together with 40 countries, with different sizes and also different levels of scientific infrastructure, ERGA has followed 6 basic principles to form their structure. While the consortium aims to keep scientific excellence and the state-of-the-art for sequencing genomes, this should happen using a distributed infrastructure across Europe, with the aim of increasing geographic and also phylogenetic representation. ERGA is also committed in being inclusive under all social aspects and to treat data following fair and care guidelines, while looking into conservation and societal needs when deciding about the prioritization of species for genome sequencing. In order to attain the principles and to guarantee representation, ERGA established a council of countries with 2 representatives per country, and also scientific committees that mirror those of EBP, representing the entire genome establishment workflow. ERGA has also established Science Plus committees that support the consortium in following legislation and ethical principles, growing the engagement from scientists and citizens, and also to become a truly inclusive network. The first large endeavor from ERGA is the ERGA-pilot, which is undergoing since last year and is sequencing about 100 species that represent almost all countries in Europe and a large taxonomic range. In parallel to the pilot, a set of institutions connected to ERGA has applied in response to a Horizon European call to create and manage European nodes for the Earth BioGenome Project and the International Barcode of Life. For that purpose, the ERGA consortium has joined forces with the barcoding community in Europe, represented by ERGA-BIOSCAN-Europe, forming the Biodiversity Genome Europe project. The BGE project has therefore both the barcoding and the genome streams. The ERGA stream aims at generating about 500 genomes for critical biodiversity in collaboration with the ERGA community of scientists and for biodiversity hotspots across Europe. While the number of genomes is limited by the budget of the project, this process will allow ERGA to establish an initial distributed sequencing and computational infrastructure that should be expanded in the future. It will also form new networks of biodiversity sequencing, biogenome applications, and citizen science in connection to genomics. Deliverables will also include new standard operational protocols, toolkits, and training to increase Europe's capacity for assembling and analyzing genomes. Finally, the project will consolidate ERGA as the European EBP node. Very importantly is the fact that this project is connecting ERGA with the barcoding community very early on, so that both communities can find their synergistic points and work together generating genomic resources for supporting actions towards biodiversity conservation. ERGA is therefore a biogenome project that will help speeding up the use of genomics and biodiversity in the continent. But more than that, it's also an umbrella network in biodiversity genomics across Europe and will establish distributed computational and sequencing infrastructure, apart from the network of scientists that got formed and that is exchanging knowledge and expertise. And finally, also connecting, supporting, and being supported by the many genome projects that are in place or getting started across Europe. With this, we expect that ERGA will expand and interact even more with the other nodes from the EBP consortium and with the international consortium itself. I thank you very much for your attention.